exciting to be part of this symposium, of course, with all the exciting talks. So sorry for any repetition from yesterday, but I'll expand some more. So I have a project called Hydrons, or Hydrozoa in Sweden. And yes, I'm a PhD student at the University of Gothenburg and would like to be considered. But we focus on Anthoathecate diversity in Sweden. Oh. So Hydrozoa taxonomy actually used to be an active field in Sweden. During the 1700s, Linnaeus and Forssell described several taxa. These are just a few examples that I show you here. And the field continued to be active during the 1800s with several monographs and other contributions. But during this whole time, the focus was usually only on the benthic diversity, so missing the pelagic stages and thereby also missing a lot of the diversity, potentially discriminating against Anthoathecates, as many of them have very small and similar looking polyps, like several of the hydractinids, for example. And after the 1800s, this field kind of came to a halt in Sweden. During the last century, the only targeted survey that's been done was a 1-week-long workshop led by Dale Calder in 2010. But then, of course, they've been covered to some extent by more general phonistic surveys as well. For example, a huge one in the 1920s and 30s led by Leonard Jägerskiöld. And then homage to that one led by the Swedish Taxonomy Initiative, a 4-year survey. But again, they completely excluded the pelagic stages, which is also showing in the numbers for Anthoathecates that they've been underrepresented. In the 4-year survey, for example, they only saw 13 species. So currently, this leaves us at 73 species ever recorded in Sweden, but over 20% of these haven't been seen ever since they were first recorded or even described. And with no one working on Hydrozoa in Sweden, our national records have become outdated with old synonymies accumulating and misidentifications hanging around, no one to validate new records. And we know from work with DNA barcoding and integrative taxonomy done outside of Sweden that morphology doesn't necessarily tell the whole story. Cryptic speciation is common in Hydrozoa. And integrative taxonomy has also proven useful to link previously unpaired life stages, which could come in handy here because several of our species we don't know the full life cycle for, even for very common species here, like ICAR-CMFRI, where we haven't seen the polyp. So currently, Hydrons, sorry, maybe I missed, no. Hydrons is the only group working on Hydrozoa in Sweden, and we can do this thanks to funding from the Swedish Taxonomy Initiative. They were born in 2002 after a commission from the Swedish Parliament to actually describe all multicellular species here and distribute information about them. But the project's also dependent on the expertise from my external supervisors, Louise Martel and Aino Hosia. You heard Louise tell you about worm-filled jellies yesterday, and you should probably go check out Aino's poster on gelatinous zooplankton in Norway. They've been studying hydrozoans in Norway in their projects Hypno and Norhydro for many years. And through Hydrons, we in Sweden can kind of tap into their expertise and build on the finds they have from there here. And this is really essential because even though Thomas Dahlgren, my main supervisor here in Sweden, is a knowledgeable taxonomist, he doesn't specialize in Hydrozoa. And apart from us, the main group, we also have collaborations. To the far left in the picture here, you see one example that you met this morning, Johan Soto Angel and his project NOAH. Here's from a sampling effort this summer. So there's quite a few parts to this project. A central task, of course, is to provide an updated national species list with robustly confirmed species. So we're collecting fresh material from diverse habitats to create a geotag reference collection, along with barcodes that will be freely available, CO1, 16S, and ITS. If you look in bold now, you'll see that there's very few barcodes from Sweden, and they have some issues possibly with misidentification. And combining these data, the morphometrics, the biogeography, the genetic distances, we will explore the diversity and evaluate dubious species and describe new species, etc. But also, aside from this integrative taxonomy part of the project, we're interested in how well eDNA is at gauging the diversity compared to traditional methods. So we'll look at this through data to metabarcoding. To find, to maximize the diversity we find, we concentrate on the marine areas. So mainly the northwest coast of Sweden, where we have the highest salinity and the highest diversity. And around in this area, we sample both shallow localities by hand and go out on ships to sample deeper localities by dredging and doing grabs and taking plankton nets. And to see how the eDNA compares, we take water samples at multiple depths to see which part of the diversity is represented where. Can we, for example, see the benthic community in the water column or even at the surface? So this is where we are now. We're just 10 months into the project, so it's early day, but I can give you a brief overview of the results. We've finished our first sampling season, which has included 2 week-long stays at Kristineberg Center, one of the 2 marine stations run by the University of Gothenburg. And I spent a few additional weeks at the other station teaching and maybe sneaking some samples from the courses there. And a little by chance, I got to tag along as the Swedish Meteorological and Hydrological Institute did one of their monthly samplings on board research vessel Svea, going down along the west coast and into the Baltic and back, taking plankton along the way. So yeah, the black spots in the map here is where we have samples from this far. And along the way, we've had a few new records for Sweden, including Velella Velella, or By the Wind Sailors, that actually beached around both the marine stations this fall. So that was quite exciting to finally have them. But our nets and dredges don't select for Anthoathecate, so we have other curious finds as well. 2 that we've seen most this summer are actually among these new finds, the Narcomedusa solmaris and Mugia atlantica, or Mugia atlantica, that we've heard a lot about in several talks now. Interesting to hear from Maria that they didn't find them in the offshore water. Otherwise, the current that comes in here would have been a good source. But yeah, and we've had 1 instance of Helgesidakari that's also new for Sweden. But back to the Anthoathecates. We have at least 25 species this far, but all the material is in process, so there's potentially more. But the ones we have include several that are very rarely reported. One example is Eudendrum cnidophorum, which is described from here, but not seen in the field since 1907. We have sequenced this, and this far it does nestle within Eudendrum, but it doesn't match a barcoded species. But we need to look at more markers because there are some that could be confounding that aren't, we've only looked at CO1. And 2 very different examples are Hybocodon prolifer and Pachycordyle. Both of these are quite common species. We've seen them a lot, but there's more or less no records in Sweden, likely just because no one's been looking. And we also have data supporting cryptic speciation. So these are a few that we will look closer at. And first out is the genus Euphisa, where we will build on results from Hypno and Nordhydro in Norway together with NOAA for looking at the species that are present in our region. And Euphisa aurata is very common in the plankton in both our countries. But barcoding suggests that it's actually 2 species under this name. And this is supported further by morphological differences that strictly follow the genetic differences. And besides these clusters, there's 2 more species in Sweden, Euphisa tentaculata and Euphisa flammea. And Now, only Euphisa aurata has a described polyp stage, and since that's now a species complex, it's a bit unclear to which one that to which medusa that polyp belongs. But we have molecularly confirmed polyps for 3 of the 4 species in the region. And together with Yuan and Project NOAA and others, we're looking at the family Tubularidae, including the hydroid Tubularia indivisa that you see down in the right corner here. This might also be a species cluster. There's molecular data suggesting this, but however that pans out, we will do species fact sheets for the species present here. If you look in the Swedish database now, you'll see that there's no pictures, no descriptions, so there's really low incitement for people to report them if they see them in the field. They won't maybe know what they find. And finally, there's Cordylophora caspia, an invasive species that's probably a complex. If you look in bold 9 now, you'll see that it might be up to 4 species under this one name just lumped together. And currently we don't know which ones of those we have in Sweden, but we have them over a huge salinity gradient. So we're curious to see. Right now, the only record we maybe have is a potential first lake record from Sweden, but we need to confirm that molecularly first. But the plan is to go sampling next year along the East Coast to see if we find any difference in the, any molecular evidence for differences along the salinity gradient, or if it's all the same actually, to be able to understand how they disperse and potentially manage them. It's, I think, a reasonable first step to see who is here and who spreads where. And that's all I had for now. Thank you very much for listening and for this opportunity.